In this lesson, we will look at the different factors affecting the rate of water uptake in seeds, and these include the seed composition, seed coat properties, cell water potential, soil physical properties, and overall seed morphology. The process of imbibition of dry seeds is dependent on various factors, namely the chemical composition of the seeds, seed coat permeability, cell water potential, soil physical properties, and seed overall morphology. Let's first discuss how composition of seeds affect water imbibition. After pollination and double fertilization, the zygote and the primary endosperm develop and mature. Seed maturity can be divided into two stages, the physiological maturity and the harvest maturity. Physiological maturity represents the end of dry weight accumulation of seeds and is marked by the detachment of the funiculus from the ovary wall. During physiological maturity, the seeds can have 20 to 50% moisture content per dry weight, depending on the species. In rice, physiological maturity is reached 8 to 14 days after pollination and subsequent fertilization. A seed that reaches physiological maturity is already capable of germination, but it cannot be stored for long periods of time because the high water percentage in the physiologically mature seed won't allow arrest of metabolic processes in the seeds. Harvest maturity, on the other hand, is when seeds already underwent a controlled process of desiccation, bringing the moisture content down to 10 to 15% of dry weight of the seeds. The low water content reduces the metabolic activities and allows the seed to remain viable during extended periods of dormancy. By harvest maturity, glucose units have already been polymerized into starch. Polymerization of approximately 10 to the fifth glucose units ensures its insolubility. Proteins have been packaged in protein storage vacuoles where they form deposits that are soluble only in saline solutions or aqueous alcohol. And the lipids have already been partitioned into oleosomes or oil bodies, and the minerals have been bound as crystalline phytates. Seeds at harvest maturity typically have very low water potential. Water potential is a measure of the energy status of water in the seeds, or the availability of water to participate in chemical and physical processes. Seeds at harvest maturity have water potential as low as negative 400 megapascals. The very low water potential in mature seeds is attributed to the low osmotic and metric potential due to adsorption of water molecules by cell components. This low water potential in mature seeds allows them to rapidly imbibe when water is available. Now, among the various chemical components of seeds, proteins are principally responsible for water imbibition. Proteins exhibit both positive and negative charges that attract the highly charged polar water molecules. In contrast, carbohydrates in the form of starch molecules have little or no impact on imbibition. The uncharged structure of starch only attracts water at a very acid pH or after high temperature treatment of seeds. Different seeds are going to have different requirements for water during imbibition. For example, soybean imbibes 2 to 5 times its dry weight, while corn only imbibes 1.5 to 2 times its dry weight. This is due to the higher protein content of soybeans compared to corn. You can see here that soybean contains 37% protein, which is way higher than the 10% protein content of corn. Now, in terms of the water requirement of the basic structures of a seed, species like rice, barley, and wheat have twice the requirement for water in the embryo compared to the endosperm because of the higher proportion of proteins in the embryo compared to the endosperm. Aside from the chemical composition of the seed coat also affects the rate of water imbibition. Recall that the seed coat is a protective structure derived from the integuments of the ovules surrounding the embryo. The seed coat is composed of one or many layers of dead cells and often contains phenolic compounds such as tannins, polyphenols, and proanthocyanidins. All these polyphenols, combined with the accumulation of dead cells and other chemical components, make the seed coat impermeable. A good example of seeds that are impermeable are those of a lotus plant. Lotus seeds are considered hard seeds. Lipids, tannins, and pectic substances make the seed coat very hard, preventing water and air to enter and degrade the seed. So if you want to grow lotus seeds, you're going to have to make a small incision into the hard seed coat to allow water imbibition. In some seeds, there are additional structures in the seed coat that allows for easy entry of water. The micropyle, for example, is the remnant of the integument of the ovule that serve as an entry point for the pollen tube during double fertilization. In the seeds, it is present as a thinner tissue or microscopic hole in the testa or seed coat. The micropyle aids in the entry of water into the seed during imbibition. Aside from the micropyle, we also have a structure known as the hilum, this structure that you see right here. The hilum served as the funiculus attachment to the ovule. In a mature seed, the hilum also permits easy water entry into the seed during water imbibition. Other structures that aid in water imbibition include mucilages and pedicel. You already know from our previous lectures that mucilages are polygalacturonic acid polymers deposited in the seed coat. Mucilages hydrate quickly and allow for uniform water absorption across the seed surface. The pedicel, on the other hand, is the point of attachment of the grain to the panicle branch, as you can see in this picture. The pedicel is the structure encircled in green. Pedicels are open, porous structures that allow hydration of seeds during water imbibition. In most grasses, rapid hydration moves from the pedicel to the coleoptile, while a slower, more progressive hydration moves through the seed coat and into the endosperm. Water availability inside and outside of the seed can also affect water imbibition. You already know that mature seeds undergo a controlled process of drying or desiccation. This drying process increases the overall water potential of seeds. The water potential in seeds is a result of three forces. One is the cell matrix force, two is the cell osmotic concentration, and three is the cell turgor pressure. Cell wall matrix force is the ability of cell walls and membranes of intracellular inclusions to attract water molecules. What do we mean by intracellular inclusions? These are structures inside the cells that are bound by cell membranes. The more intracellular inclusions, the higher the total cell wall matrix force. In these panels below, you can see the different intracellular inclusions in seeds. Panel A shows protein and oil bodies in Arabidopsis labeled in P or OB respectively. Panel B illustrates protein storage vacuoles and protein bodies in rice, labeled as PSV and PB respectively. And finally, in panel C, you can see here cell walls in date palm. All these structures are enclosed in cell membranes, which contribute to the cell water potential. In general, cell matrix force aids in water imbibition. Aside from the cell matrix force, we also have the cell osmotic concentration affecting water imbibition. Cell osmotic concentration refers to the concentration of soluble compounds within the cell or seed in a larger sense. The higher the concentration of soluble compounds, the greater the potential to attract water molecules. Recall from your biology class the principle of osmosis. So if you look at this illustration here, when you have two systems separated by a differentially permeable membrane, you can expect the water to move through the membrane from an area of lower concentration to an area with higher concentration. In mature seeds, the drying process makes the seed a highly concentrated system. This concentrated system aids in water imbibition. Lastly, we have cell turgor pressure. This refers to the pressure resulting from the restraining force of the cell wall. When you have high turgor pressure, it means your cell is already turgid or expanded from water entry. High turgor pressure retards water absorption. On the other hand, low turgor pressure means there is not enough water in the cell. That means there is still room for the cell to absorb water. Low cell turgor pressure, like the condition of the cells in mature seeds, aids in water imbibition. Soil physical properties are also important determinants of seed water imbibition. We have here as examples a sandy soil and a clay soil. The clay soil absorbs more water more vigorously and retains them longer than soils with high volume of sand. In effect, seed water potential has to compete with soil water potential for imbibition to occur. That means your seed will imbibe water faster when you put it in sandy soil just because the sand will not hold the water. Now, during imbibition of seeds on the ground, the initial difference in seed and soil water potential is reduced as the seed imbibes water. So after some time, you see a dry area surrounding the seed. Now, soil hydraulic conductivity replenishes the water surrounding the seed, allowing the seed to continue imbibing water. Last but not least is the overall seed morphology as a determinant of water imbibition. In particular, we will look at the effects of seed morphology on seed-soil contact. There are three seed traits that can improve seed-soil contact. First is the presence of structures that increases the number of pathways through which water may be absorbed by the seed. A good example of this is the mucilage. Number two is the altered seed coat configuration. Textured seed coats are more likely to have higher seed-soil contact than smooth-coated seeds. And lastly, we have seed size. Smaller seeds have higher surface area to volume ratio than large seeds. The higher surface area permits them to have access to greater amounts of water than large seeds. To summarize, water imbibition is the rapid uptake of water by the seeds that results from the hydration of seed storage polymers. The extent to which water imbibition occurs is dependent on the composition of seeds, seed coat permeability, cell water potential, soil physical properties, and overall seed morphology. Proteins are principally responsible for water imbibition. Seed coat structures such as mucilages, micropyle, hilum, and pedicel also aid in water imbibition. The availability of water to an imbibing seed is controlled by both the availability of water within the seed and the external environment under which the seed imbibes water. Internal seed factors include cell matrix force, cell osmotic concentration, and cell turgor pressure. External factors include the soil physical properties and overall morphology of seeds that determine seed-soil contact.